forms, it is taken up again by the mosquito, and the cycle continues basically. Okay, you can just see how further down here, and the mosquitoes again, goes around, and it changes in mosquitoes to oocyte, and then again, the female that causes the disease, the male is, is just there for giving genetic variety. So this is called spores, and erythrocyte outside the red blood cell. This is inside the red blood cell. So it's a very complicated cycle that takes place. And I just thought I'll throw that in for good measure. So, okay. I think somewhere along the line I lost my slide share. Okay, let me see the slide share again. Start. Okay. Okay, yeah, here we are. Okay, let's do a slideshow. And I think we were somewhere here. Let me see the malaria cycle. Sorry about that. Okay. Okay, so hopefully you're able to see this slide, and I'll set up from the current slide. Okay, yeah, and okay, I think we should be able to see the slide now. Okay, and this screen too. Okay, and malaria has been a big favorite of the Nobel Committee, and we already talked about Ronald Ross, and even as recently as 2015, because the quinine was around, and which was the plant that was stolen from South America, and quinine was planted in Southeast Asia, and was a big part of the gene and tonic. Now, Chinese medicine had a lot of herbal therapy, and a Chinese scientist who was an individual runner based on the recommendation of Chairman Mao, worked on isolating a chemical which became artemisinin, which was dramatically useful for Chinese yew plant, YEW plant. And so Madame Tu, she was a Nobel Prize winner in 2015. Okay, and let's see, I think I'll stop share. I want to make sure that I'm sharing the correct thing. Share screen. Yes, it should be okay. Yeah, we should be in business. Okay, moving on. As we mentioned earlier, several vitamins have been recognized for Nobel Prize, and starting with the Dutch name Eichmann, discovered the vitamin which had to do with beriberi, which was occurred due to a polished rice. And the man who coined the word, his name was Casimir Funk. He was a Polish scientist, and he had isolated varieties of active factor. Okay, and he word said these were chemically related to amines, so that's how the word vitamin came about. At the same time, in Americas, a disease called pellagra was occurring, with skin rash and organ failure, and that was related to vitamin deficiency of niacin, vitamin B1. At the same time, a British scientist named Hopkins, Dr. Hopkins, had discovered that just by giving animals the essential casein, protein, and carbohydrates, they were unable to grow. So there has to be some other factor that was responsible for growth of the experimental animals. Okay, and chickens in those cases. So that gave the concept of essential growth stimulating vitamin A and other vitamins where the idea was proposed. So he was recognized for, and he also worked on biochemistry and showed that the tryptophan was one of the essential vitamins. And later on, it was until 30s that the various other vitamins and the pigments were isolated by dissection of eye and animal eyes, primarily frogs, I think. And that showed the pigment rhodopsin, which was essential. So the vitamin A and vision, it came into this thing. And it took for several years for Nobel Committee to recognize the value of this particular discovery. Okay, now the most dramatic discovery of the early 20th century has to be insulin. And this was the connection between sugar and insulin was mentioned in the class, even in the Indian Ayurveda, and the role of pancreas was established in the Western science by MacLeod. Okay, he was a Canadian scientist, and his mentors, Fleming was working with him, and again, along with his student Collip, they were able to isolate juice from the pancreas of the animals, and were able. Well, now in those days, juvenile diabetic was absolutely a fatal that, and they were, after doing animal experiments in dogs, they became brave enough to treat a Canadian boy who was, was dying uh, from diabetes, and he had dramatic response, and the rest is history. And so Benting and Best, were the founders of the insulin. And later on, very quickly, insulin was isolated from cadaver pancreas from pigs and cattle, and that started the big pharmaceutical industry. And Eli Lilly became the big giant of animal-based insulin, the pork insulin versus calf insulin. So that was the early ways. Nowadays, obviously, that has all fallen by the wayside with genetically designed insulin, which was again a big discovery, big invention of Genentech. Okay, and these were obviously, they were a well-deserved Nobel Prize. Now, somewhere along the line, Collip was left out. So MacLeod, Best, and Benting got the prize, and the gentlemen that Benting and Best were, they shared the rewards with their young student, Collip. But so there were actually 4 people, who were involved, and okay, and later on, and there were some other people who were involved in insulin research as well. The insulin structure was determined, later on by crystallography by Frederick Sanger, okay, and Dorothy Hodgkins. You can just see here, this is Frederick Sanger, and the younger days and older days, Dorothy Hodgkins, and Israeli a scientist, Yellow, who discovered the radioimmunoassay for insulin. So there were several people who are involved in the further work on insulin. Okay, and so we talked about Sanger, and who's probably the only unique person who has won 2 Nobel Prizes for crystallography. Okay, now sometimes the treatment can be, Nobel committees and science can get things wrong. As cancer was obviously the priority, and while working on cockroaches, I think Danish or Dutch scientist, who are named Johannes Fibinger, was working with cockroaches and was looking at the stomach content and was able to see organisms which were spirochete type. And he put two and two together that this was responsible for cause of cancer. And they were working with rats and roundworms that were responsible for stomach cancer. So he could experimentally thought he could induce cancer in healthy rats using roundworms. So this was considered a great triumph of experimental medicine. And he had also worked on rats which were fed vitamin A deficient diet, so associated, so again, the whole concept was a little on the shaky ground. Now, obviously, the cancer. Now, at the same time, a Japanese scientist named Katsuburo Yamagiwa had reproduced cancer by painting coal tar on rabbit ears as early as 1915. But his work was considered undeserving because it's it's already well known, because the little kids who work as a chimney sweeper, they get cancer, so it's not anything new. But the scientific theory that was proven by experimentally inducing carcinoma was totally ignored. And again, perhaps because he was Japanese, he got the snub. So Johannes Fibinger got the prize for medical parasitology. I'm sorry, I think I was the cockroach. No, so he was that the roundworms had a lot to do with cancer. Now, obviously, the whole finding fell to the wayside. Now, at the same time, the treatment of syphilis and the illnesses that occurred, especially the dementia paralytica or this thing was pretty scary. And the idea was that when you get fever, that the organisms, can die. So this idea was extended by inducing artificially fever. Now, how do you induce fever? Okay, you inject people with malaria parasite, because they knew how to treat malaria, but they did not know how to treat syphilis. So if by creating artificially malaria, you might be able to. So this is like the experiment you can just see on the slide as to how they're injecting malaria parasite and causing the temperature to rise. And obviously this was the temperature rose up to almost to about 105, and several people died from it. But it was still the mental illnesses that can occur were so scary that this was considered a groundbreaking discovery. And the guy named Julius Wagner Jarung was given the Nobel Prize in 1927 for treatment of syphilis. Now, obviously, that all fell to the wayside with the discovery of salvarsan, which just followed a few years later. Okay, but again, this was considered, so he must be the only psychiatrist who got the Nobel Prize, not to mention four. Now, at the same time, the technologies, electricity was considered, cornerstone, and to record the impulse of the cardiac was, required effort to how to amplify the signal. So, Dr. Since the days of Galvani, it was known that this thing. So Einthoven, was a Dutch physician. He was managed to amplify the signal that could be recorded from heart, and was able to record the cardiac impulse. And then this is, just to give you basic cardiology thing as to how the impulse starts at the top on the atria, which is the right up chamber, goes to the middle through three pathways, and then divides into varieties of tiny little branches through the ventricles and stimulates the heart. So these are called left and right bundles. So it breaks out into, so it's like the master control. And if there is a scar tissue up here, occurs either because of heart attack or aging, then you need a pacemaker. This node, the impulse center, like the nerve center, or that starts, and then it goes to the relay center and goes on to back forth. So there is problem here, then the heart rate slows down. These beats at a rate of about 70 on an average at rest, 70 to 100. These beats. Now, Mother Nature has a variety of backup system that even if this is destroyed, you will not die, but the impulse will slow down, but will continue. And so, the heart will be less efficient. So this is the basic cardiac cycle. Now, for whatever reasons, he discovered it. Instead of putting it A and B, he gave the name P and QRS. So this is the little bump that you see is the P wave, which shows the contraction of the top chamber of sinus node. And this, the bigger impulse, the ventricles are bigger. So this is the other side. Okay, so that's the basic EKG cycle. So just for 101. Okay. Moving right on too. So, and we already talked about the roundworm type of thing. And at the same time, there are a lot of suspicion that went on as to that the microbes had a lot to do with cancer. Now, if it's schistosomiasis, some of them were actually correct because the parasite that occurs in Egypt due to contamination and parasite that can get through your body from your foot and can affect the human bladder. That does cause bladder cancer after a long several decades of infection. Liver fluke, which is another parasite, can cause bile duct cancer. So those are legit. And even later on, the H. pylori, which is the dramatic discovery that the tiny microorganism was now all along in the early 20th century was considered the H. pylori, but that ulcer was caused due to stress. But Barry Marshall and Warren, they did self-experiment. They drank the H. pylori cocktail, and sure enough, they got gastritis and ulcer. Peyton Rouse had done research on sarcoma, and had discovered a virus, and that was a Rouse sarcoma virus. So that got the scientist on the wrong chain of thought that the cancer had something to do with virus. I already talked about the coal tar virus. Okay, and this was the huge equipment weighing several thousand 100 pounds of Dr. Einthoven's EKG equipment as to amplify the current by putting in salt water bath and amplifying the current through what was known as a galvanometer, and was able to record the impression as to how the limbs are immersed in salt water to amplify the current. Okay. Now, again, along with penicillin, the most important discovery of that time has to be blood groups, because the blood transfusions have changed the think of modern medicine, and this was discovered almost with concept of antigen antibody by taking the blood groups of taking different samples from different people, and this was done by a guy named Karl Leinsteiner, who was a physiologist, pathologist, and initially did work in Germany, and later on, for personal reasons, I think he moved to the United States and worked in New York, and he was able to collect samples from various of his colleagues, and is able to prove this is how you can just see that if you have A and antigen B, the blood would coagulate antigen antibody reaction that takes place, and if you don't have any antigens of either A or B, and then you don't form antibodies, so this is A, B, and if you have antibodies against A and B, okay, so that's the blood group O that takes place. So this is, the basic principle of antigen. So, okay, so that the first blood transfusion was carried out, at Sinai, New York in 1907. So O negative is considered the universal donors, because again, okay, if you don't have antibodies, type of thing, and so so they consider, so that's one way that's considered very rare blood. Okay. And he had also discovered later on, he did other blood groups called M and N and RH blood groups. So, and that was a change that had a huge impact on. Okay. Now at the same time, a German scientist named Otto Warburg had worked on cancer cells metabolism as to cancer. Says can be caused by varieties of other causes, So, and he worked on, the cancer cells also need nutrition. So he worked on the concept of that role of as the cells have respirations, the cancer cells needs, metabolic cancer is a metabolic disease. So it's a different theory was proposed type of thing, and he was even though he was Jewish, he was continued to allowed to work in Germany, and perhaps partially because Hitler himself was deathly afraid of cancer, so that's why the Nazi Germany was ahead in in passing anti-smoking laws. Okay, and now his ideas where he probably was a little bit over the top, so to speak, that it was not all due to because he had a hard time believing the concept of genetics being involved even after the discovery. So he continued to support his own ideas of cancer being metabolic disease type thing. But now it turns out that he perhaps was not totally wrong, and because it takes multiple factors, the cellular metabolism and the networking effect has a role to play along with genetics, because a lot of times you find that there are mutations that are there apparent in cells, but still not everybody gets cancer. Okay, so that was again an equally, so both concepts had some valid. So cancer metabolism is quite complex, as we'll talk about as we go along. Okay. At the same time, we just said the neurologists had their own thing as to whether it's a chemical impulse or electricity. So brain electricity was suspected as far back, in the early 19th century, and the idea was further proposed, by a guy named Hans Berger. Similarly to heart, he was able to come up with the recording electroencephalography, and that changed the diagnosis of epilepsy dramatically, and later on, even though this was not a Nobel, he did not get the Nobel, a Canadian scientist, okay, they came up with the concept of how the brain named Penfield, while stimulating, doing surgery for epilepsy, cutting out the area which causes epilepsy, he was able to map the representation of the human body into the brain. It's called the Penfield maps. This was done, in Montreal. And so this is how the whole body is represented. The more sensitive areas are represented wide and broad, like a tongue and eyes and hands and fingers. Okay, and similarly to Golgi and Cajal, the controversy continued between the electricians and the chemists of the brain as to what was important as to reflex arc of electrical impulse, which was well known from the electric eel versus chemicals. Okay, and so this idea has continued, the various schools, but it turned out both of them were right in the end, that the brain uses both the mechanisms, so electrical discharge as well as chemical, the neurotransmitters, and now initially the other transmitters, only one or two, as acetylcholine and dopamine, were discovered, but now there are at least 50 to 75 neurotransmitters have been discovered, including the one for morphine or narcotic receptors type thing that's induces the pressure type thing. So, and various British scientists, Lord Sherrington, who put the concept of reflex arc through the spinal cords as to how your knee kicks out when the doctor taps your knee tap with a rubber hammer, and the electrical discharges were again proven as to how the shock treatment works, and so they were able to, so the both ideas came into a foray. Now, interesting theory as to how Otto Levy, who had a dream about trying to show, and this is the famous experiment that as to how chemicals have a lot to do with. So he did an experiment on isolated frog heart. They took the juice from one side, simulated the nerve, and put it in the other denervated heart, and was able to slow down the heart rate. So this was the classic. He had a dream at one night, and he wrote it down quickly and went in the night to the lab and performed the experiment. This is the famous dream sequence of Otto Lowy, and that obviously showed that yes, there was something in the that was proven to be acetylcholine, which was secreted by by the vagus nerves. Okay, so this was along with the British neuroscientists, they were recognized for the discovery. Okay, and as it happens quite often in discovery, that people who were working, in the United States, Whipple and Minot, they were working on a different type of secretions that are produced from the stomach juice, So it has a lot to do with what we call Whipple factor, and has to do with the various essential vitamins that were secreted, i.e., a vitamin, B12 later on was isolated from, and the parietal cells, and the role of iron and something called the intrinsic factor had to do with, so they did the work on, on these things, and they were assisted by a able assistant named Frida Robbins, but she was ignored as to this was suspected, back in as far in the 19th century, it was suspected there was something in the liver, and Frida was the first author working with Dr. Whipple, in Rochester, and so, but again, for whatever reason, she was, and the hemoglobin pigment was converted into bilirubin, so that's how they work with, that was done mostly. All of the work was American, and along with that, and so this lady had as many as 20 publications, and but she was not recognized for her thing, and and later on, Dr. Minot had diabetes, and he was one of the first recipients of insulin. And he had worked with another well-known physician who became the founder of occupational medicine, Dr. Ellis Hamilton. He had shown the occupational hazards of lead and TNT, and along with that, it's varieties of carcinogens. So you can say that she was the founder of occupational medicine, as we know it. And we already talked about the various vitamins. A Hungarian scientist named Albert von Szent-Györgyi had discovered vitamin C, and he was the inspiration to various Hungarian scientists, including the recent Nobel winner, for COVID vaccine. Okay. At the same time, the bleeding and other stuff was discovered in Wisconsin, in Madison, Wisconsin, and Dr. Henry Peter Dam discovered vitamin K. So the vitamins were a big favorite. Okay, obviously, so this again elaborates some of the more this thing, and and obviously the importance of vitamin C in scurvy was known to sailors even in the 17th century. Okay, and the Hungarian scientist had worked with paprika and had isolated various chemicals, which were converted into ascorbic acid. Okay, and so Györgyi was a big name in chemical research. Okay, and the Coumadin story I've told many times, I won't repeat that. Okay, and now the embryology was researched by a German scientist named Hans Perman. Okay, and again he was assisted by a PhD assistant named Hilde Manigold, and she was his able assistant and did the very delicate transplant experiments on embryos. Okay, and demonstrated that as you transfer cells from one side to the other, the axis formation of the body, the north-south or left-right axis, is formed and evolves. Okay, so she was, was working on, baby frog or newt experiments. Okay, and it showed that, that the certain cells were very extremely important, and there was the cell organization that took place in what we know for this thing called the blast cells, and other things that she had worked on, the basic embryological development. Unfortunately, she died due to a gas explosion and was never recognized for her work. Now, with the rise of Nazi Germany, the chemical industry was very strong, and various dyes were investigated as an antimicrobial agent. And Gerhard Domag had worked on varieties of dyes, including azo dyes, which were, a product of IG Farben. And after testing thousands of chemicals, he had come up with one idea that they named Frontosil, and which was had a strong antimicrobial property. Okay, and was inactive in, and so the mice that were infected with Streptococcus, dramatically responded. So antibacterial. Now he was so dramatic in fact that it was a red type of dye, so he named it Prontosil rubrum and patented it as Prontosil. And he used it on his own daughter who had strep infection. Now, unfortunately, he was not allowed to accept Nobel Prize for several years by the Nazi government. Similarly, another scientist was working on sex hormones and isolated the well-known hormones, estrogen, progesterone, testosterone. He was again denied. And he worked on, urine from animals, distilled about 4,000 gallons of urine, okay, and was able to isolate sex hormones. And again, so this is, and again, it is notorious for abuse by athletes, and it was used as an attempt to cure gay males, okay, and it's again the hormone therapy is a big industry type thing. So, okay. And testosterone again is the big money maker for drug companies and a pseudo advertisement. And at the same time, curing renal failure was the Dutch scientist named Dr. William Kolff, had worked on it, and with the geriatric equipment using juice cans, auto parts, and sausage filters, had come up with the concept of dialysis, and had later on moved to the United States and had donated his equipment to hospitals and never tried to patent it. And that was the start of dialysis. I don't think he was ever recognized for his work, Okay, and now the third dramatic discovery, along with insulin, blood groups, has to be Dr. Fleming's discovery, and Fleming and which was as early as 1929. Okay, and the penicillin mold that was created was sold for 14,000. So that was the groundbreaking discovery. As to he was he was going on vacation and had left his growth on the windowsill uncovered, and somewhere along the line somebody on the lower floor was working, and it was a particularly hot day in London, and so it is possible that the spores went up through the window and settled on the petri dish that he was working with, and there was no growth in where the mold had settled. And Dr. Fleming's reaction said, "Gee, that's funny." Okay, and then again he wrote a paper for it in some obscure journal, but was left. So again, the animals, his experiments were rediscovered and by Fleury, and then he came across the paper that was published several years ago, and again, and then that just finally, the discovery stayed quiet for almost 10 years, and during the war there was a mad race to mass manufacture penicillin, and finally Chain, who was an Australian scientist, they traveled to the United States to look for where naturally occurring fungus can be found, and it was found in a cantaloupe in Peoria, okay, by a microbiologist. So it's a very fascinating story, and the mass production was cranked up by the American pharmaceuticals, and okay, and so that was and that just changed the course of the war, okay, and Winston Churchill was treated for it, and and there was a dramatic risking as to the American companies were able to patent it before the Brits, and now in the middle at the same, so the penicillin has to be one of the third biggest discoveries. The fourth one has to be the rise of genetics and the modern genetics, because Mendel had proposed that in the earlier century, but Thomas Morgan, working with fruit flies, and his assistants, who were equally capable and went on to were able to show that, because otherwise the genes was a concept and chromosomes were known during cell separation had occurred, and based on the mutations that they were able to meticulously document in the fruit flies, they were said that their genes were actually a biological entity, and the effect of the genes, that they were able to show that the chromosome are present within the cells and have a genetic material, which again, the DNA code, had to wait for another 20 years. And so hereditary transmission did occur, and they were able to mate the fruit flies and show that the red eye or less a wing, and there actually was things, and they were able to prove something came to be known as a linkage correlation. So Thomas Morgan, and so this was the start of what we call as a classical genetics. Okay, one of his students, Hermann Mueller, went on his own and was, to create mutations. Obviously, waiting for nature to show mutation was hard. So he started using, his brilliant idea was to use X-rays to create mutations, and the work that was done in 1920s. And again, so that he alerted people to the dangers of radiation, and that became very relevant in the aftermath of atomic bomb. Okay, and now again, he was a socialist and moved from United States earlier to Germany, and from Germany, with the rise of Nazism, moved to Russia. But after the dissolutions of Lysenko and Stalin, he came back to U.S. And so he had his share. But again, so in 1945, it was somewhat politically motivated that his work was recognized for, uh, for creating mutations and the hazard of radioactivity, and it was recognized right on the and with the hydrogen bomb, it became, and he had written a letter along with Russell and Einstein with the danger of nuclear testing. Okay, and again, this was various scientists, including physicists and Linus Pauling, were very active in now. At the same time, metabolism required research, not Marie Curie, but Curie, and the Curie is where showed what is known as the glucose cycle, as to how glucose is metabolized into various chemicals and forms lactic, which again stops the, which builds up, and if you're running, the lactic acid builds up. They meet the cycle of lactic acid metabolism that takes place in the liver that was established by the Curies. Okay, in 1947. So so they were, and again, Gertie Corey faced discrimination as to that she was never given a job at Johns Hopkins because she was considered hiring a couple would be a distraction and considered nepotism. So she was not given a job, and she was just given a sort of a low job as a lab assistant, opening the German correspondence. Okay, and later on they moved to St. Louis, and she was able to work along with him. And again, so this was one of the stories as to how women face discrimination. Now, this has to be shared with you as to one of the worst Nobel Prizes was a Portuguese scientist named Antonio Moniz. Again, to cure mental illness, he had come up with the concept of a lobotomy as to introducing a sharp object through the nasal cavity and destroying part of the brain to make people. So he did publish work on experimental on 20 patients in 1926, working in Lisbon, and they were cured of their mental illness. They showed some slight improvement, but others stayed same. So this was he proposed. Now again, they had side effects which were ignored, such as incontinence. They became like a zombies and memory loss. Okay, and again, they did a very primitive, crude surgery, and this was taken up with enthusiasm by an American neurologist named Friedman. Okay, and they continued doing operations in, on several hundred people, and with very dubious results. And the most well-known example was Rose Kennedy, the Kennedy family daughter, who was, who became like almost like a vegetable and had to be institutionalized. So this is leucotomy was for treatment of depression, in widespread use, became popular for a short time, and when it was proven that it has side effects were a lot more worse, and so that has to be one of the worst Nobel Prizes of lobotomy. Okay, and including ECT. So this is Dr. Friedman doing surgery and pushing in an ice pick from the nose. Okay, and became, so they did experiments on chimps earlier. And again, so this was a dramatic demonstration of how medicine can go wrong. Okay, and then again, this is, I'm going to jump over one slide. Why medical students change their major. This is the so-called Krebs cycle, which is how metabolism takes place from pyruvate to citric acid cycle, as to how it undergoes various chemical changes, and that's was known as, this was discovered as by Dr. Hans Krebs for the citric acid cycle, and as to how the metabolism takes place. So it's something that you just memorize on the day before examination, and then forget it very quickly. Okay. Now on the tails of penicillin, the one fungus worked for for the gram-positive organism. The more serious organism was tuberculosis, so-called acid-fast organism, and so this the hunt starches for looking for fungus. So fungus was discovered in the soils, and in the farmland in New Jersey by a grad student called Albert Schatz, and he did most of the work, while his mentor, Dr. Waxman, was away, and he was deathly afraid of catching tuberculosis himself. So his grad student did the work along with various physicians in Mayo Clinic. But behind the student's back, Dr. Waxman had a contract with Merck, and was instrumental in getting financial gain. And Rutgers stood behind him, and Dr. Schatz screened thousands of the fungi, actinomyces, that were discovered, and was able to show that yes, that the particular, streptomyces growth was successful in controlling the tuberculosis growth. But he was shortchanged, and he went to the courts, and finally he was given some compensation, but never recognized. And Rutger stood behind their principal investigator, Dr. Salman Waxman, and he was considered the founder of tuberculosis and was even awarded Nobel Prize. At the same time, some of the other bad discoveries that were there were, obviously was useful at that time, because malaria was rampant and killed thousands of people. And so DDT was, Dr. Paul Hermann Mueller was awarded Nobel Prize for discovery of DDT. And at the same time, Rachel Carson had already alerted people about the dangers of widespread DDT. Okay, moving on, the physiology prizes were given for discovery to Dr. Kanda, because the Germans were buying up a lot of adrenal glands from carcasses, okay, in the United States, because they wanted to secrete a chemical that could help their pilots. So that inspired the research for see what they were up to. So the research was done by Dr. Kandel at Mayo Clinic and associated. So they were able to isolate adrenaline from the adrenal cortex, and it was used for, at the same time, other physicians were looking for cure for severe rheumatoid arthritis, and cortisone was given and as a sample, and fortunately they tried out fairly large dose of cortisone, and the rheumatoid arthritis patient were dramatically improved. Okay, and so that was, and cortisone became the workhorse of varieties of illnesses, as varieties of uses. It has wide-ranging effect from the brain all along with and the whole hormonal cycle, that the entire endocrine system came into existence. And obviously, if you have deficiency of endocrine hormones from cortisone, you get Addison's disease, which Kennedy suffered. Okay, and by this time, the other people had isolated thyroxine, which we already alluded to earlier, and the master control pineal gland, which has a lot to do with the growth hormone. So finally, there was the endocrine coming into its own. The only vaccine to get the Nobel Prize until the most recent, obviously, the mRNA vaccine, was a guy named Tyler. Okay, and now this is somebody who may recognize. This is the great Walter Reed. For him, then Walter Reed Hospital is named. He was working in Cuba along with a Cuban scientist, Dr. Finley, and this was considered the first principle of, the Baconian principles. They did the research on reproducing yellow fever in test subjects. So they were asked for volunteers. They were given as many as $100 in and gold, and varieties of people volunteered for it, including some nurses and his assistant named Lazar. Okay, and Dr. Walter Reed never volunteered himself, and then so that was and he got the recognition for curing yellow fever. And Cuba recognizes Dr. Finley's contribution because he had originally done a lot of the work on this thing. So it's an interesting human interest story. And it took until 1950s, to isolate the virus for yellow fever and create a vaccine for yellow fever. And the yellow fever vaccine can be pretty toxic, and you can get due to the virus, which is grown in chicken embryos. It can cause severe neurological damage also. So the varieties of people got some various side effects from the vaccine, and including some of the vaccine that is still used for travel to Africa, and can have some serious side effects that can occur. And recently, one of the British reporters who got the vaccine, has documented all of his illnesses, and the documentary was done on BBC. Malcolm is slightly sick, so that is an interesting story about and now the vaccine has been modified again, so that probably not as toxic as it used to be, but that's still the Dr. Tyler's vaccine, which was recognized by Nobel Prize in 1950s, is still being used for travel to endemic regions for yellow fever. And yellow fever was a pretty serious illness during the early days of America in Philadelphia. Okay, and now okay, and again now the genetics discoveries were becoming very popular, and the Spanish scientist Dr. Severo Ochas in Kornberg worked on the DNA and RNA working with E. coli, and they were able to isolate various enzymes and able to work on the basic mechanisms of how DNAs and RNA work. Dr. Kornberg worked with his wife, who was equally able scientist, and at the same time, Dr. Ochas, who was a Spanish scientist, worked on various other mechanisms of RNA, and and it has had an able assistant postdoc who worked with him. And now, obviously, this is again the most famous discoveries of the 20th century. Has got to be the DNA structure based on the famous photograph 51 that was done by Rosalind Franklin, who was shared by her supervisor, Dr. Morris Wilkins, with Watson and Crick, who traveled down from Cambridge to University College. And based on that, they came up with the idea of the double helix structure and announced it at the pub. And Linus Pauling had closely come. And this is the basics of genetics type thing. As you can see, that this new human cell has nucleus. Inside the nucleus, the chromosomes are there, the telomeres at the end, the junction point, and the telomeres are at the end. And DNA is wrapped around the histone protein and the classical double helix with the sugar phosphate backbone and the A, C, G, and T, the nucleotides are on the inside. The mistake that Linus Pauling had made was he had put, the nucleotides on the outside, which have the negative charge and would be repelled and could not combine. And well, the Watson and Crick put them on the inside based on Rosalind Franklin's thing. So that's the groundbreaking discovery in 1953, which got this turned classical genetics from Morgan to the present day of molecular basis. Okay, a lesser known scientist named Dr. Morris Hillman, and was instrumental in making about 20 or 30 vaccines, including encephalitis and measles, mumps, all this. So he's one of the most famous scientists, work at Merck, unprecedented success in creating measles, mumps, rubella, chickenpox, hepatitis, meningitis, pneumonia, you name it. Okay, but he was still an unsung hero. Okay, Morris Hellman. Yeah. And this is the quote from Anthony Fauci that very few people had ever heard of him. Okay, and now this was a question that I should have posed, type thing. Who won the Nobel Prize? It wasn't Dr. Salk or Sabin, but the two people who had done the basic work on chicken embryos for reproducing poliovirus, Enders, Fellis, and Robbins. That's why Salk and Sabin, who did the work on it, they were a few years too late. And the big discoveries in genetics. Now genes are there, but they don't do the work. The enzymes and the proteins do the work. So one gene, one enzymes was done. The famous experiment were done by two scientists, Beadle and Tatum, and they were recognized for their work. Work on a simple organism, the bread mold, and showed that how depriving the nutrients, the particular amino acids did not grow, and so that the concept there was originally proposed by British a clinician named Archibald Gerard, was one gene, one enzyme. So the abnormalities of gene produces abnormality of the particular amino acid, the phenylketonuria. Severo Ochoa was a friend of Salvador Dali, and Salvador Dali was impressed by this. He was not a scientist by any means, but he was impressed, so he did his tribute to Severo Ochoa. So this is his famous printing of polynucleotide messengers, so this is hanging in the museum of his lithograph. Okay, and this is the famous Biddle and Tatum experiments as to how, the wild type and in the mutant gene, the enzyme is missing and it produces, certain amino acids as to how, it is created. So this was the classic experiment of creating one gene, one enzyme experiment that was done by, okay. And again, similar experiments are done in phages, type of thing. And phages are the tiny viruses that affect the bacteria. Okay, and you can just see, is this the bacterial set that is the defective phage, and the virulent phage, the so-called, and then it is transferred. And again, the bacteria, the phage is injected inside the DNA, and it causes changes, inside this thing. So transfer of portion of DNA from one bacteria to the other. So it's again, so 1958 discoveries were recognized that these some of the pioneering works. At the same time, cardiology was selective angiogram was discovered by accident. Dr. Soanes was working, and the catheter went inside the coronary arteries, and so that showed that yes, that it is possible to inject dye inside tiny coronary arteries that the patient would not die. So this was, so this was the start of heart disease. And that will come into the Nobel Prize. Let me jump along to one more. And before that, in 1950s, a German urologist had done an experiment on himself and had sweet-talked a somewhat gullible nurse into helping him to put a catheter inside his own body. And because initially it was thought that heart is so sensitive, if you inject or mess with it, it would stop immediately. So he disproved the idea that here's the famous photograph. He inserted a sterile, small, tiny catheter inside his heart, walked over to the radiology department and took an X-ray. And he was chastised by his superiors for doing a self-experiment and in different areas. And I think I had to go back to urology. And later on, working at the Nazi government, he was a war prisoner in Britain. That's when he learned that he'd won the Nobel Prize. Later on, he was rehabilitated and went back and practiced urology in Germany, never did any cardiology. So he, along with the other researchers, got the Nobel Prize, several years later, almost 30 years later, for, for coronary angiography, from urologist to cardiology to war criminal. Okay, now let's see, who did I miss? Now, two French scientists were working on genetic control of enzymes, type thing. So this is called the lac operator, because bacteria usually prefer glucose, okay, but if they're given galactose, okay, so they change certain enzymes would be active and reactivated type of thing. So this is the concept of repressor. So a negative feedback loop called lac. So this was the idea of genetic control of enzymes that was proposed by two French scientists, Francois and Jacob. Yeah, now we already talked about him. And so then we showed about the people who do the experiments from themselves. We starting from Niels Finchen, the guy who did the light therapy. Again, we already talked about, Bentig volunteered to work with, and he gave himself, so this is again a side of the policy. The tradition of CESPA experimentation is well known. Okay. Now, again, we already talked about during physics as to linear accelerators were developed at Stanford, and Ernest Lawrence was savvy enough to raise funds by using a linear accelerator for treatment, okay, of cancer. So physics and medicine went hand in hand. And at the same time, Margaret Mead and Sanger, okay, were interested in population control, and based on Mexican plants of yam, Syntax Corporation was able to isolate chemicals which would, induce hormones and progesterone. And there was a start of the progesterone pill that was tested out in the female hormone estrogens and male hormone androgens were isolated by the Germans. And this was used on a trial basis, for initially marketed for menstrual irregularities, and there was a start of the birth control pill. Somewhere along the line, that never got recognized as a very much useful research, because perhaps it was already known, and the hormones were already known, and so this was, never recognized for the work. Okay. Dr. Pincus, who did a lot of the work on the birth control pill. So it's an interesting paradox as to, and Peyton Browse, okay, now again had to wait for a long time for his recognition. And this was the Rouse sarcoma principles. Chicken with sarcoma in the breast, he removed the tumor, cut it up in small, injected it again. So how the virus can cause, so that again, led the scientist on a wrong chain for a long time. Now again, we already knew that the DNA and RNA have a lot to do with genetic code, but obviously the proteins are the workhorse. So somebody had to do the basic work, and this was the genetic code that was done by using various nucleotides and trying to decipher, the genetic code. The work was done by a Florida-based scientist, Dr. Marshall Nirenberg. He put the uracil in and this thing, and they were able to create, now the game mode as to how the genetic code was, whether it needed 4 nucleotides or 3 or 4. So various physicists were, coming up with the ideas. Remember when we talked about, the expanding universe and the Big Bang, a Russian scientist named Gamow had suggested the idea that the genetic code may have chemistry, may be related to physics. And so that experiments were done by a biochemist, Dr. Nirenberg at NIH. And then again, the work was, and he was lucky enough that he was working with uracil. So they were able to isolate the enzyme. It's the amino acids very quickly. And then the work was further elaborated by Holly and Indian-born scientist in Harbagomin Corona in 1960. So pretty quickly, the whole amino acid sequence was reproduced as the DNA codes was deciphered, and starting with Nuremberg. And Nuremberg had a low budget lab and he presented the papers in Moscow, and very few people were paid attention to it. And James Watson, who was the big honcho in molecular medicine by that time, he asked that to be present his papers next day, and then everybody else got on the mad race. And the most interesting thing that Nobel oversights, like Virginia Apgar, who presented the Apgar baby score, that was never recognized. Same way, other pediatric scientists, okay, were not recognized as well. Okay. Okay, and continued with the other scientists. Obviously, this is the classic DNA and central dogma that Crick and Watson proposed. But working with virus researchers, David Baltimore and the people, that sometimes the viruses work, in a backward way, that the RNA, messes up with DNA, and that's they updated the so-called dogma and created protein. Okay, so so that was the way of the viral etiology as to how viruses can occur and can mess up with the DNA. And that's obviously the HIV is the classic example of it. In the same way, the virus were isolated for hepatitis in Australia, and that was, the start of the other virus causing us various diseases, such as hepatitis was the big factor. And the same way, polio and other things, became a known thing. So, and again, the HIV story in 1980s is well known as to how that had a huge role. Now, even things are even smaller. The Kuru virus, which was not even viruses, there are even smaller proteins which were isolated in Papua New Guinea as to by consuming a brain that is infected with prions, with the tiny protein particles which can cause this disease, and can be transmitted. That was proposed by Gajusek, and he's probably has the only dubious distinction of ending up in jail because he had brought in some boys from Papua New Guinea to I think Carolinas, where he worked with in his lab, after his earlier research, and he was guilty of abusing children. So he ended up in jail and served for almost several months, and finally died somewhere in Europe. Okay, and again, so now I think I'm going to skip over some of these. These are much more esoteric and are not of interest. Okay, and again, Hubel and Wessel had worked on the basic neurophysiology. Okay, and as to how the eye impulse is transmitted, and so there was some of the earlier work in neuroscience. Okay, as to that worked with varieties of eye reflexes and asked neurons where the signal is coded in the brain as to the ocular predominance. I think so. And they've isolated how the signal is transmitted, light signal, the Hubel and Wessel cat experiment. So initially, if the cat's eye is patched, that the memory formation doesn't take place. So the whole idea of how the vision is transmitted, that was discovered by Hubel and Wessel. Okay, as to how the closed with at birth, the eye is closed, so how the interpretation does not, memories cannot be formed and vision does not develop. At the same time, women kept getting short changes, and even in the early 70s, Barbara McClintock, I'm sorry, 1940s, working on corn or maize, Barbara McClintock had proposed the concept of that the genes can actually do not need to be transmitted. They're called jumping genes that occur, and he had labeled it transmitted genetic elements. The word that was coined was transposed genetic elements was proposed called jumping genes. But her ideas were not accepted for almost 30 to 40 years. And eventually she was proven she was right. The mobile genetic elements, that there is more to it than simple DNA to RNA. Okay. And various other people have worked at Cold Spring Harbor Library. So Cavendish Lab is big in physics. Cold Spring Harbor Laboratory in Long Island has huge, a lot of geneticists have worked with there, including, other people who are lesser known and not well recognized. Margaret Voigt, okay, who was Del Bruco, who had worked with phage therapies, his assistant, okay, and again, so their work was, less recognized. So there's a huge list of geneticists, starting from Morgan, Mueller, Barbara McClintock. So this is like a huge list of genetics. Switching gears, the heart disease was, cholesterol theory was proposed way back, it may have something to do with it. But the LDL receptors, which are associated on the liver surface, how the cholesterol that is taken from food is bound into the liver and the cholesterol cycle. The LDL receptors were discovered by two guys named Brown and Goldstein, and they were recognized how is to the ApoE protein and binds the cholesterol esters on the liver. LDL particles are taken up and metabolized inside the liver and are recirculated. Cholesterol is necessary for formation of hormones side. And so this idea, this was, now working on the other end, other side of the world, endopharmaceuticals was working on antibiotics and had discovered various modes and worked, and at the same time this mode had some side effect on lowering cholesterol in animals. Okay, this was discovered by a chemist who was working with endopharmaceuticals, type thing. And he had proposed that this might be useful, but Endo was not interested in pursuing this particular mode of work. And so this whole library of Endo's work was bought by Merck, and Merck continued to work on this a way to block these receptors, and it's called HMG co-inhibitors. And there was a start of the statin therapy, and this drug that they came up with was Mevaline or Mevacor. So the initial drug, but for Endo, was somewhat toxic, and Merck worked on different type of chemical structures, and and there was a start of the statin therapy, and that revolutionized the treatment of heart disease. On a British scientist named Dr. Blake, was responsible for discovery of varieties of drugs, beta blockers, the Inderal, Integumet, Cinemet, AZT. So he can be said the giant of chemotherapy. Okay, and Gertrude alone was a poor Jewish woman from New York City. She worked as a lab assistant at Hunter College, and then later on, enrolled in PhD program but never finished. Okay, and she worked, on discovery of various, okay, so she shared the Nobel Prize along with, I'm forgetting what her actual work was, okay, and the AZT therapy was initially obviously very expensive type of thing, but that changed the treatment of HIV dramatically. Okay, antibodies, this was the big saga that the Brits still have the sort of egg on their face that they had, Milston and Kohler had discovered something called the monoclonal antibodies, how mice are challenged with antigen, and their spleen cells form something called hybridoma, and based on that they've selected varieties of cells and had formed something called the monoclonal antibodies. Now they didn't get around to patenting it, but they had shared the technology or the procedure with Wistar Institute in Pennsylvania. Okay, scientists a lot of times share information, and Wistar Institute, they managed to patent it and made tons of money from it. And obviously the British government, including Maggie Thatcher, was upset, as to how they missed the boat as to commercializing the inventions. And the challenge was because once they tried to patent it in Britain, because the British laws are such that once the method was published in Nature magazine, you cannot patent it anymore. So let's say they kind of lost the race by a few months, Okay, and at the same time, there are other people who have done work on something called growth factors. They are a very human interest story. Rita Levi-Morgantini was a scientist and was working during the Mussolini era. And her father did not favor education. She convinced him to allow to continue. Okay, and during, obviously she was not allowed to work in the lab. She continued to work in her own lab in the bedroom, by boring chicken eggs on growth factors in embryos, and communicated with Dr. Hamburger at St. Louis University, came back to St. Louis and worked on something called growth factors in embryos. and was recognized for her work with Nobel Prize, okay, along with Stanley Cohen. Okay, and now the stem cell research, that was done by Murray and Thomas. This was the bone marrow transplant that was carried out on the west coast at University of Washington as to how patients receives hemopoietic stem cells are harvested from the donor, they're collected, they're separated and returned to the patient. And after, so this is the principle of bone marrow transplant, which revolutionized the treatment of leukemia and other things. Okay, and are the founders of bone marrow therapy and later on for organ transplant as well. Ion channels, which we again talked about, that thing. So that was again one of the bigger discoveries. Okay, so I'm not going to get too much into details of the of it. Now, the other well-known, sorry, I think there was something here. I had a video that I think I decided to eliminate it for. Okay, the most important discovery, which is the staple of forensic medicine and NCIS, and Carey Mullis was a chemist, and he was back in the 70s, he was not above indulging in hallucinating drugs, and he had come up with the concept of what came to be known as PCR, or the long name stands for polymerase chain reaction, as to how DNA fragment can be denatured at high temperatures and cooled off, and by adding various DNA primers like the A, C, G, and T segments, and the fragments are multiply exponentially, and that's called the PCR reaction. By heating and cooling, by adding fragments of other basic nucleotides, you can see how DNA is separated first, and by adding other things, further DNA copies can be manufactured, from 1 to 2 to 2 to 4, and so forth, so DNA section and amplified by adding code fragments. So that's called the polymerase chain reaction that takes place, and he patented the technique. Okay, but then after publishing papers, he had arguments with remuneration with the corporation that he worked for, called Cetus Corporations. Okay, and then later on, I think he'd probably done a little bit of too many drugs, and he became AIDS denial, climate change denial, astrologer, and so again, so it just goes to, just because you won a Nobel Prize doesn't mean you're all stable. So the heating, denaturing, and cooling it off by lowering the temperature and multiplying. The cycle keeps repeating. That's called the polymerase chain reaction. Okay, and the prion diseases were further outlined by the misfolded proteins, which we talked about by another scientist named Stanley Prusner. So varieties of diseases, not just Kuru, but Creutzfeldt-Jakob disease, amyloid disease, some misfolded protein, which I'm sure Peter will talk more about it when he does his class. Now, we talked about the Nobel Prize in the nitroglycerin, but what's the real mechanism of nitroglycerin as to nitrogen enzyme, how it produces the vascular relaxation? Okay, that took a long time for it to be discovered as to how the relaxation takes place inside, the cellular mechanism works, by the nitric oxide. Okay, the blood vessel, what the role is inside of the nitrate in causing platelet aggregation, thrombosis, relaxation of blood vessels at various stages of cardiac cycle, that takes place. And that took, until very recently, it was discovered finally by the true scientist, one. Okay, let's see, I think, I have to go forward on this. It was discovered by Turkish one of the few Turkish scientists, Farid Murad, along with the two others, by the nitric oxide as a signaling molecule in cardiovascular system. Okay, now again, the Nazi legacy continues with only one that will concentrate on Eric Kandel, who was there from a Nazi exile to Nobel Prize. Okay, so we'll play this video if I can.
Doing science is a very satisfying way to spend one's career. Pick a problem that is really important and try to tackle it in a very, very serious way. Don't do a trivial problem. It's better to make small progress on something really, really important than to push a trivial issue further ahead. My scientific questions often come from things that I have personal interest in. I went to Harvard and majored in history literature because I wanted to understand what happened to me in Vienna, 1938, when Hitler marched in. I wanted to see how, civilized people could become so brutal and anti-Semitic. While I was writing my dissertation, I met a famous psychoanalyst, and he said to me, if you want to understand motivation, if you want to understand what happened to you, you're not going to do it through history and literature. You've got to really study the mind. You've got to study why people do things. You've got to study psychoanalysis. I had no science background whatsoever. I took physics, I took organic chemistry, took the courses necessary for medical school, and I went to medical school with the idea of becoming a psychoanalyst. I came along at a very good time when neuroscience and psychology, which were worlds apart, was beginning to come together. I was one of many people that contributed to that. There were people before me that already started the way. But I took advantage of the fact that this was a new direction that was opening up. And there were certain experimental systems that were particularly conducive to that. And many neuroscientists of my generation were scared to work on behavior because they thought it was too complicated. And I, being a psychiatrist, that didn't bother me in the slightest. So I learned how to study behavior and worked out a simple reflex that I could study and see what happens when an animal learns something. And I found that what happens is that the strength of synaptic connections changes. The nerve cells communicate with each other at specific sites called synapses. And these synapses are changed by learning. With certain kinds of learning, the synapses get stronger. With other kinds of learning, they get weaker. Well, I think working out the first cellular mechanisms of learning and memory is not so bad. And I'm quite proud of the fact that I did this. Way beyond that now, the field, which was very small when I came into it, is now gigantic.
Okay, so this was, and so he worked on aplasia, small ROC organism and animal rather, and long-term potentiation of memories that takes place with acetylcholine. So that was Eric Cantle, and he's really a true, well-rounded scientist. Some of his books that are there about the culture and art are just phenomenal. So I would strongly recommend for those who are interested in reading. Now, with regard to obviously the Human Genome Project was funded by the Congress and was going to take 13 years, and they were able to finish it, I think, in about 8 or so in 2001. The biggest discovery of the 20th century has to be the Human Genome Project. So it was published simultaneously in Nature and Science magazine with Craig Venter, who was a private company, and NIH. Okay, so this was the start. Okay, and now again there are some other genetics continued to about Sidney Brenner, who was a South African physicist, come biologist, who worked on phage therapy, and in UK and workhorse of genetics, called C. elegans, a little tiny worm. Okay, and that's everything has been known about it. The whole genetic code has been deciphered, and it's the workhorse of and the neurology, and all the neurons have been mapped. So it's like people have learned a lot from C. elegans. Okay, now they make MRI is the third technology that has been very popular with Nobel Prize and has changed medicine in the study of neuroscience dramatically. And the people who have made it useful for medical use are Lauterberg and Peter Mansfield, another scientist named Damodian who had worked on it separately, and somewhere along the line, even though he had the patent on it, he was never recognized for his work. Okay, so it's just we'll have to wait for 50 years to find out. How does an MRI work? This can be of interest to most of us.
MRI is a medical imaging device commonly used to examine the soft tissues of the human body. Let's meet the machine. The key components are the magnet, radio waves, gradient, and a computer. Our bodies are made up of 60% water, and water is magnetic. Each of the billions of water molecules inside us consists of an oxygen atom bonded to 2 hydrogen atoms. We know it as H2O. Small parts of the hydrogen atoms act as tiny magnets and are very sensitive to magnetic fields. MRI stands for magnetic resonance imaging, and an MRI scanner is one of the main diagnostic tools that doctors use to examine inside our bodies. The first step in taking an MRI scan is to use a big magnet to produce a unified magnetic field around the patient. The gradient adjusts the magnetic field into smaller sections of different magnetic strengths to isolate specific body parts, for example, the brain. Normally, the water molecules inside us are arranged randomly, but when we lie inside the magnetic field, most of our water molecules move at the same rhythm or frequency as the magnetic field. The ones that don't move along the magnetic field are called low-energy water molecules. To create an image of a body part, for example the brain, the machine focuses on the low-energy water molecules. Radio waves move at the same rhythm or frequency as the magnetic fields in an MRI machine. By sending radio waves that match or resonate with the magnetic field, the low-energy water molecules absorb the energy they need to move alongside the magnetic field. When the machine stops emitting radio waves, the water molecules that had just moved along the magnetic field release the energy they had absorbed and go back to their position. This movement is detected by the MRI machine, and the signal is sent to a powerful computer, which uses imaging software to translate the information into an image of the body. By taking images of the body in each section of the magnetic field, the machine produces a final 3-dimensional image of the organ, which doctors can analyze to make a diagnosis.
So you can just see that it's math and highly complex technology. Okay, and and we already talked about Marshall and the people about the self-experimentation for H. pylori. So this is, the standard thing that, but this is, I think I'll just skip over this for the time being. Okay, and this, okay. Now, again, the saga of the Nazi era continues. The Maria Capucci had worked on the stem cells and that thing. So, and he was, he's an even more gut-wrenching story that he was, he was left as an orphan in post-war Italy, and it took, survived on the street as a 6 or 10-year-old, and finally united with his mother. Fortunately, they had connections in Philadelphia, in Pennsylvania, so he was sent to Pennsylvania with his uncle, and then obviously was a brilliant student and managed to get PhD and worked his way up to Nobel Prize. Okay, so it's a very dramatic story as to how he survived, the horrors of war as a young child. Okay, and this was that earlier work on post-war orphanage, discovering the principles of gene modification on stem cells. Okay, but the human interest story is even more interesting than the research per se. The HIV was a big problems, and was a massive health problem in 81. Okay, and it took was a lot of controversy that went on as to how, and this is the timeline, as to how many millions of people were affected, and the search for virus was a difficult virus research that took, and the fight went on between NIH and the Pasteur Institute, and so those were the people who actually were eventually able to isolate the virus for HIV, and along with that, the French people, Mongolia, and Francois Barré Simosi, and the man who was making a lot of effort to be included, Robert Gallo from NIH, he was kind of late because he had used the French virus that they had given him as for test samples. Okay, compared to this, Schatz and Selman streptomycin research, the telomeres, where finally, the postdocs started getting enough credit, and Carol Grinder, who was a young Australian postdoc working with Elizabeth Blackburn, was recognized for work in telomeres, which are kind of a little end segments of chromosomes. And as the chromosome ages, they wear out, and telomeres and have to do a lot with the aging process called telomerase, and became very popular for a while. And her mentor, Carol Blackburn's, the guy named Jack Szostak, who has done a lot of work on RNA chemistry and origin of life. So they shared the Nobel Prize, in 2009. Okay, so we have got a few more years. Now, this is again another interesting human interest story. This is Ralph Steinman, who is the only person who actually received a Nobel Prize after he had passed away. Okay, because the Nobel Committee was not aware that he had died, and he had done a lot of work on immunity. Okay, but so they was, finally the committee decided that, well, we were not aware that he had passed away, so he was given, and he had worked on developing he had pancreatic cancer, had tried to develop cocktail and immune therapy for his own illness. So it's a very interesting human interest story. Okay, and here we have Nobel Prize 2010. Okay, that was given for IVF. It was given to Edwards, and for how the medical infertility, as to how they have been able to, when conception fails, the eggs were dried from the women, okay, were implanted in a test tube, fertilized outside, and this is the thing that and when it was considered, there were a lot of protests from the people as to how playing God. But again, now millions of kids have been born with in vitro fertilization, and so that was recognized for Nobel Prize in. Okay, and other people who had worked on in vitro fertilization at the same time, were the creators of Dolly, Sir John Gurdon, okay, who had done the ultraviolet radiation and had isolated nucleus and had done the cloning of the baby. Okay, so that was considered, the groundbreaking discovery. At the same time, because of the stem cell controversy, a guy named Yamanaka, who had worked on varieties of genes and carefully with virus, genes were implanted, and they tried to reduce what came to be known as Yamanaka factors. So 4 essential factors were necessary for creating any stem cell, any regular skin cell into stem cell. So it's just like, any stem cell can be made into a skin cell, can be transformed into liver cell, heart cell, brain nerve cell. So this came to be known as a Yamanaka factor. And so this is, this was one of the groundbreaking research on induced pluripotent stem cells. So this was done along with Thompson. Okay. Now Nobel Prize, has been, MRI research has been popular, starting from physics, and it gradually morphed into, biology. So it's the incorporation of various disciplines, from physics to pure physics and to chemistry to medical sciences. So it's a long saga of how the invention have been used for human benefit. And the varieties of brain images, starting from from X-ray images to computerized tomography, which was received a Nobel Prize in 1958, to MRI scanning and MRI angiography, and we already talked about the positron, which was discovered, in 1939. So these are, is to X-rays are just standard, just shows the bone. CT scan shows the brain, but not good details. It can show bleeding and etc. MRI shows good details, and small bleeds, so a lot more. Angiography, which shows the blood vessels, and the PET scanning shows the metabolism, okay, and of cancer cells, and so, but it's a very expensive and complicated technology. So varieties of technologies are used. Okay, moving on to a few more that are left, type of thing. Okay, let's see. Okay, parasitic diseases, like the river blindness, filariasis, because that's still around, we don't see it here. Madam 2, UU, which we already mentioned about during malaria, artemisinin discovery, and so various other parasitic diseases were recognized, late, but finally were okay. And autophagy, where the cells actually, proteins are destroyed as proteins are formed and destroyed in the body type of thing. So that was discovery by, in Japan. Okay, I think I'm going to skip some of these. The GPS, the place cells in the brain, that were the European discovery. Okay, moving on to circadian rhythm, our body clock, that was recognized in 2017. Okay, moving on to the changing of monoclonal antibody therapy. Okay, or the Jim Ellison and Honjo discovered. This was a groundbreaking thing, which is called the CTL4 and PD1 therapy. Okay, so let's see now. Let's cover this. This is of interest, I think, as to how tumor cells have certain proteins on the body and how their T cells can, work on, by blocking them, we can activate the T cells, and so that would, selective program destruction of a malignancy that takes place. So that is one way of doing. Another more that Jim Ellison worked on was called CD24. This is one of the proteins again on the cells, as to how antigen presenting cells, it is involved in tumor cells. By blocking these at the correct level, T cells are activated in causing tumor destruction. So these are some of the T cell proteins which were studied as far back as in 1990s, and developed into a specific chemotherapy for cancer. Okay. Okay, a Nobel Prize in 2017, we already talked about, I think earlier, the circadian rhythm, which is the internal body clock that takes place, so-called period gene and timeless gene. So, and that's, so that was one of the recognition. 2019, okay, that was about the oxygen. Now, moving on to the most important prize that, again, the dramatic discovery, there was, is about CRISPR. So I'll show you the CRISPR video for.
The CRISPR-Cas9 system is a tool for cutting DNA at a specifically targeted location. The technique has already revolutionized gene editing, but scientists are always looking for new possibilities. So what else can CRISPR do? Since being discovered in a bacterial immune system, CRISPR-Cas9 has been adapted into a powerful tool for genomic research. There are two components to the system: a DNA cutting protein called Cas9 and an RNA molecule known as the guide RNA. Bound together, they form a complex that can identify and cut specific sections of DNA. First, Cas9 has to locate and bind to a common sequence in the genome called a PAM. Once the PAM is bound, the guide RNA unwinds part of the double helix. The RNA strand is designed to match and bind a particular sequence in the DNA. Once it's found the correct sequence, Cas9 can cut the DNA. Its two nuclease domains each make a nick, leading to a double-strand break. Although the cell will try to repair this break, the fixing process is error-prone and often inadvertently introduces mutations that disable the gene. This makes CRISPR a great tool for knocking out specific genes. But making double-strand breaks isn't all CRISPR can do. Some researchers are deactivating one or both of Cas9's cutting domains and fusing new enzymes onto the protein. Cas9 can then be used to transport those enzymes to a specific DNA sequence. In one example, Cas9 is fused to an enzyme, a deaminase, which mutates specific DNA bases. eventually replacing cytidine with thymidine. This kind of precise gene editing means you could turn a disease-causing mutation into a healthy version of the gene, or introduce a stop codon at a specific place. But it's not all about gene editing. Several labs have been working on ways to use CRISPR to promote gene transcription. They do this by deactivating Cas9 completely, so it can no longer cut DNA. Instead, transcriptional activators are added to the Cas9 by either fusing them directly or via a string of peptides. Alternatively, the activators can be recruited to the guide RNA instead. These activators recruit the cell's transcription machinery.
Okay, so that's a very complicated thing, so I don't think too many of us would be interested in it, but the basic idea is as to how it's a precise gene editing, and that was, the two ladies got the Nobel Prize for that. Okay, and again, now this is, the most recent Nobel Prize was given for temperature and the caspacin, as how temperature is perceived in the body, as to how the signal is transmitted from when you feel pain, and the same way for touch and pressure. So that was okay. Now moving on to the next thing, the last prize was 2022 was for Neanderthal gene. Okay, Svante Pabo. So that was the isolated of the Neanderthal genome, and Denisovan genome was the Swedish scientist Svante Pabo. Catalina Carico. Okay, this is the last one. It's the final slide, promise. Okay, and this is
our immune system and new drugs. The goal of a vaccine is to stimulate our immune system to precisely target a particular infectious agent, such as a virus. Traditional vaccines use a weakened virus or parts of a virus to stimulate the immune system when injected. But the new vaccine instead uses mRNA to act as a template for how to build the virus part, which is made by our own cells and later triggers the immune response. mRNA vaccines are much quicker to produce and less expensive than traditional vaccines. It's how we were able to develop effective vaccines against COVID-19 so quickly.
So we are finally finished the whole circle up to 2000, from 1901 to 2023. So it's a very long video, but I'm sure that those of you who are interested will enjoy it. Thank you, folks. I think we will see you sometime in future.